Okay, so for the last part on this chapter is actually on protein-protein interactions. So, well, I will basically proceed with protein bioinformatics, but probably in next week. Because protein-protein interaction is mostly, or you could more or less associate or correlate with the protein bioinformatics, which is on docking. But let's take a look at what are the protein-protein interactions. So we know that, well, just a bit on systems biology. Systems biology, okay, represented such as this is actually very wide, okay. So from single protein to systems biology, it's actually kind of like it involves a lot of interaction, either from protein-protein or protein substrate or protein ligand. Now, what are protein-protein interactions? So it means that protein that works together, okay, forming multi-complexes to carry out the specific function. And this is what happens with protein because usually it works together. There are proteins that basically has a sole function, but usually it's because protein has multi-purpose or multi-functions, so usually it involves another protein, okay, and they work together. So in terms of experimental approach on how to study protein-protein interaction is via histohybrid, remember histohybrid, okay? Or we can use affinity purification from your chromatography, or usually have to be by the addition of mass spectrometry, okay? So first and foremost, okay, for this kind of protein-protein interaction, you need to have a target protein. You cannot just use an unknown protein, just want to explore anything. You have to have a target protein because without which then it wouldn't be a protein-protein interaction. You have to have something that has been defined. Now, for the interactions, so usually you have to identify it. So you can do both ways, okay, experimental or computational. But a lot of times, okay, it has to be, of course, confirmed by the experimental. A lot of times people would start with computational first because it's the easiest to do. Okay, it doesn't involve a lot of money. Yeah, to probably involve a lot of time, okay, but not as much time as compared to experimental. So let's have a look at them or compare them side by side. Okay? So in terms of experimental work, okay, you need to do some of the experimentals using X-ray crystallography, okay, NMR spectroscopy, or mass spectrometry, okay, in terms, or sometimes using tandem affinity purification. Okay? Or you can use immunoprecipitation or histohybrid or microarrays, okay, which is similar to microarrays for genome sequencing but for protein. And well, for each and every one of them, it will take a lot of times from, not to dampen your spirit, from 1 year to more than 3 years, 4 years, 5 years, more than that, unless somebody has established it beforehand. Alright, and apart from that, it's also quite expensive to conduct all this, okay, because all these chemicals, solutions, reagents, needs are expensive, simply said. For the computational, the cheaper side, and cost effective, and probably won't take as much time as this, is whereby when you have a set of computer or hypercomputer, if you can have, which would be much better, and you can basically more or less play around or do a data mining, basically do a data mining from either genomic data or structural data. So do you know that, if you don't have a complete protein sequence of certain organism, you can actually use a genomic data, and basically then proceed with the protein bioinformatics. You can actually do that, because this is done by phylogenetic profiling. So remember your phylogenetic tree. So from there, actually from the profiles, we can determine and we can select for the best protein to be used. So for the genomic data, you can also get some gene context, gene fusion, and also symmetrical evolution, to help you with choosing the correct protein and also the sequences. In terms of structural data, this is very important, obviously, and as we have seen from the previous video on the structural databases, so sequence profile is important. This is to build up, the model, which we will do, and apart from that, because you also need some template from there, so 3D structural distance matrix, would also be useful, and this would be provided, by established database such as PDB. Apart from that, you can also get certain surface patches. This is important for docking and binding interaction. Alright, so the steps of protein interaction network are as such. First, you need to have the sequences and the networks. So you need to know how it works by predicting interaction sites. So binding site prediction, basically should be done to understand network evolution, to understand changes in protein function, to predict the protein interactions, and of course, manipulate protein interactions. So sequence-based prediction, okay, could also be done, okay, whereby you could predict interaction site and motif. So this is an example, okay. So from interaction map up until interaction set, okay, so you could get motif, okay, discovery after you remove the domains and homologous regions, and you could get a predictive or predicted motifs of how the interaction would be. So this is from a paper. So if I were to explain, okay, from here to here, it's actually going to be very lengthy, so I'm not going to delve into that, okay. It's just that, okay, usually this would be the last one, okay, that people do, okay. But okay, if it has been established, what you can do, okay, is that you can just study, okay, pick up, okay, on interaction and how it interacts, okay, in terms of the pathway, and then you can just determine, okay, of the homologous region, what overlaps between them, okay, and then you just remove the domains and homologous regions, so you will actually get, okay, certain proteins and how it basically function and behaves or interact. Second step is, of course, predicting the protein interactions. So whether interactions of orthologs or interologs, okay, so another one for you, okay, phylogenetic profiles, and also domain-based predictions. So for orthology-based predictions, so I asked you in the previous video on what are the differences between homology, paralogy, and orthology. So you should know them by now. And looking at this, I hope that it would jog back your mind. Okay, so all of them are homologous. Okay, so orthologous, okay, which means that the same sequence, okay, from different organism. Paralogs, okay, from the same organism, okay, but there are differences, okay, in terms of the function or even sequences. Right. So we are focusing on this orthology, okay, which means that, okay, certain protein, okay, which probably would interact, okay, similarly, but they actually originates or are available in different organisms. So, okay, you need to more or less, okay, go into the phylogenetic profiles for that, okay, and see how, not say see, okay, and then analyze, okay, how they actually interact. So again, I'm not going to explain into details how this is done, how this is interpreted, because you are going to be very confused later on. Okay, but okay, usually, okay, it would basically comes back, okay, or basically related to the domain-based prediction. So domain-based, okay, it would be very useful if you can go to some of the domain structure databases, okay, and find on the predicted or reported interactions of domains of interest, if you know that your protein have domains. Okay, so from there you can try to deduce, okay, how they are interacted, okay, or how they interact with one another. So the third steps, okay, is the sequence and network evolution, and this is done via either duplications or interactions or interaction loss or interaction network evolution. What do I mean by that? Okay, for example, duplications. So as the name says, okay, I'm sure everyone understands what duplication means, which means that you duplicate something, okay, from the original, okay, you duplicate. But what happens is that after duplication, so let's have a look, okay, at, for example, the chromosome of here. This is duplicated region. So once you duplicate the region, okay, so after duplication, what happens is that the chromosome is just a bit more lengthy, okay. I would say the length would have increased, okay, but then what has been duplicated is only this region. That's it, okay. So later on over the year, okay, during duplications, after it has been duplicated and duplicated, okay, you may see that there are variations in terms of the sequences. They are no longer alike, even though they have been duplicated. So for example, okay, after it has been duplicated for a long period of time, okay, you can see that some interactions have been lost or some interactions would have been gained and so forth. So duplications and interactions, okay, work, okay, either, okay, whether it is lost or interact, okay, because, okay, when you see that when gene is being duplicated, okay, sometimes, okay, whatever, not say sometimes, okay, the most occurrence, okay, is basically interaction loss, okay, but, okay, usually if you were to see some interactions build up interaction, it would basically take some time, okay, but what you see, okay, mostly is actually on the duplication loss or interaction loss, okay. Now for the interaction network evolution, so this is actually very complicated. So this is from a journal, again, I'm as you can see, from 2011, so I'm not going to go into details to explain how they come to this conclusion, okay. So what I would summarize is as such is that they have to determine first and foremost when the application takes place, how the protein interact, and the paralogy relationship. So when they rewire the interaction, they would see that what is being lost and what not, so that they could see what are the new interactions now. So you can see that fraction of the shared interactors, previously by right, it should be 100%, but again during the rewiring it may be 50% or it may be just 0%. None of them is similar to the original. So the last steps for protein interaction network is, of course, alignment, and this is done by a local network using multiple alignment or global network. Now the difference between them is that in multiple alignment, your results will be as such. So you can get your multiple alignment in different types of parts, or in terms of different parts of the biological processes. For example, different metabolisms, as you can see over here, and so forth. For global alignment, so this is actually complicated. I'm actually taking an example from 2008 because the thing is this, if I were to explain this in physically, then it would be much better, but explaining online is actually quite hard because usually I would need to draw some diagrams. But the simplest thing, or I would just try to summarize is that for global alignment, so usually you would have to use a tool that could basically compare different protein interaction from different organisms and make sure that it could be normalized. Simply said. So for network alignment, it is actually the future. And basically how it could be used is that because without network alignment, usually you would know whether your protein-protein interaction is in the correct pathway or not, or your prediction for the protein-protein interaction is correct or not. Because again, prediction is as good as prediction is without being confirmed by experimental work. So what I would like to summarize on the protein interaction network, without going to go into details too much on it, because again this is online. So for the evolution, mostly it is done via comparative correlation. So mechanism, of course, is still unknown or unproven. Again, you can prove it, but via experimental. Approaches exist to integrate and model network analysis within the context of phylogeny, and the outlook is that you could combine interaction site prediction with network evolution analysis. So there you have it on protein-protein network interaction, the simplest that I could summarize.